Hello, everyone. Thank you for joining us today. My name is Liza Moscovice, and I'm a member of the Cost Action Lift, and I'm happy to present our speakers today. So our webinar is a special collaboration with Lift, which is the full name is Lifting Farm Animal Lives, Laying the Foundations for Positive Animal Welfare. And this is an EU-funded cost action project that provides frameworks to include positive welfare in farm animal welfare assessments. And the action began in November 2022 and will continue until November 2026. So we're in the last year now. And currently we have over 200 participants from more than 38 different countries represented. So it's been a really active, great networking experience. And within LIFT, there's a sub-working group called Bridging Disciplines, where we aim to bridge interdisciplinary perspectives and methodologies and use those to help inform our understanding of positive animal welfare. And so that's the reason that we have this seminar series. And we'd like to thank especially the Animal Welfare Slack workspace for collaborating with us and providing this platform for this series. So now I'm happy to introduce today's speakers, Drew Altschul and Bernhard Voelkl. Drew has an interdisciplinary background in animal cognition, comparative psychology, and evolutionary anthropology. And he is a lecturer in psychology at Newcastle University, where he's involved in several big team science projects which conduct cross-lab and cross-species studies to better understand the evolution of cognitive and behavioral capacities. And Bernhard has a broad background in animal cognition, behavioral ecology, and evolution, and he's conducted research on a range of animal taxa. And since 2024, he's been based in the Veterinary Public Health Institute at the University of Bern, where his research focuses on improving welfare for biomedical animals and animals used in biomedical research and improving reproducibility in science. So for the framework today, we've asked each speaker to talk for about 20 minutes, and we hope to have time at the end for questions and discussion. Along the way, if you have questions, please feel free to already type them into the chat so we can get the discussion session going quickly at the end. And also please remember to stay muted during the talks. And I think with that, I will pass it over to our first speaker. And let me stop share.
Great. Thank you very much, Liza, and thank you, Christian, and thank you all for being here and having me to speak about this. So, yeah, my name is Drew, and I'm going to talk to you today about on behalf of many primates, which is a big team science approach to studying primate cognition and behavior. So, first, I just want to talk a little bit about a couple of challenges that we have in animal cognition and just animal behavior research more generally. So firstly, how reliable are the effects in animal behavior and cognition research? How do abilities map onto phylogeny? How do environmental variables influence cognition? And how are species differences in performance structured across tasks and species? This is quite specific, so I think we can also put a bit more broadly and rephrase it as: one, how can we most effectively assess animals' abilities? Two, how do animals' abilities evolve? Three, how do they develop? And four, how are they structured? And so turns out that from our perspective, my perspective, answering these questions requires large and diverse sample sizes, pooling of resources across labs and across sites, and an infrastructure to support these kinds of studies. So this brings me to many primates. So what is many primates? It's a big team science project founded in 2018. Big Team Science, or BTS, is when many researchers get together to address research questions that we couldn't reasonably be able to answer on our own. And we're founded on principles of transparency, so that means keeping our data, our materials, our procedures all available and transparent to anyone who wants to look at them. Openness, which is more about culture. So in these projects, we try to be inclusive and welcome contributors from all around the world and from any stage of their career. We're also founded upon the ideas of democracy, and that we have a sizable membership, and when we make decisions as a group in terms of what direction we're going to be taking, we gather the opinions of the entire membership, and all these sort of dovetail with open science principles more generally. Again, open data, open materials, and pre-registration. And again, almost all basically of our work is pre-registered. And so all of these things come together to address issues of replicability and reproducibility in science. issues of sample size and bias issues, lack of generalizability, and inconsistent methods. So for instance, a project that we might want to look at is how generalizable a cognitive phenomenon is across species. Now, I want to talk a little bit about the kind of research design and the state of the field from the last, basically across the last decade, through one of the reviews that we've done as a network. So what this is we conducted a review of primate cognition papers published in 20 journals, ranging from specific journals like Animal Cognition to IJP, to more distant or general journals like Developmental Science, Scientific Reports, or Applied Animal Behavior Sciences. And we looked at a window of from 2013 to 2018. And we have 574 studies, 69 different species represented in this window. And so what we then did is we visualized these studies on this cladogram, which is what I'm showing you right here. These are all primate species, all of them. And then the gray individuals, the gray species, those are species that basically were not studied in the papers that we looked at during this time window. There just aren't any exemplars. And then if there is a point, if it appears in white, in black, then there are studies, and the size of the bubble represents how many studies there are. So let's take a little closer look now. If we zoom in on these bubbles on the right side, these are the apes, chimpanzees, bonobos, gorillas, and orangutans, so the great apes in particular. And these individuals are quite well represented. You see some representation for gibbons as well, and some lemurs higher up. But again, even here, most individual species are not being studied. If we move over here, we see a big bubble for macaque mulatta, that's rhesus macaques, and then some for other baboons and macaques. And there's a nice size bubble with Calothrix jacus, the common marmoset. But again, a lot of missing species. Move up towards the older monkeys and the lemurs. There's a nice size bubble for capuchin monkeys, and there's even one for mouse lemurs. But again, we're just not getting a whole lot of representation, it turns out. And representation also extends to where these kinds of data are collected. So we found that there are 183 data collection sites represented in the literature. The biggest five there's one in Germany, the Leipzig Zoo, one in Japan, the Primate Research Institute, and then 3 in the United States. These are the top 5. So major concentration in those regions more generally, as you can see in Europe, in the US, and in Japan, particularly strong concentration in what we would call the global north. So get down to some numbers. So the state of the field as of 2019, when we put it all together, 13 of 16 primate families were being studied. There was no research on bush babies, Galago, sportive lemurs, tartus. Only one study each on lorises, owl monkeys. And 38% of all the studies were on great apes, 184 studies of chimpanzees, 40% on older monkeys, 152 studies of those were specifically on rhesus macaques, and almost all of them were on cercopiths, that is, macaques, baboons, vervets, and the like. In terms of sample size, they ranged from very small, 1 to quite large, 481, but the median sample size was 7. 66% of all the species had a median sample size of 10 or less. And those larger sample medians were only found in studies of overrepresented species like chimpanzees or rhesus macaques. Again, size concentration, those 5 sites, they produced 38% of studies and were concentrated in the global north. Most studies did not compare multiple species. 19% of studies involved more than one species, but of those, 20% did not compare the species quantitatively. So this has problems for our inference about evolution, which is something we're very interested in. No single species should be used as a stand-in for common ancestors. So it's really important that we have a great breadth of species that we look at. It also creates problems for our ability to examine individual differences and hidden moderators, both of which are key aspects of how behavioral and social sciences are now conducted. And in the spirit of many primates, all these materials are available. You can have a look at them if you want. They're on GitHub. The code and the data and the figures that we produced from the code are all available there as well. So that's one of our reviews, but I want to talk more in depth now about kind of the experimental many primates or big team science work more generally. How does that function? How does that work? Because that's most of what we spend our effort on. So at a glance, Many Primates is made up of about 200 researchers. We've studied 60 plus species across our projects, hundreds of animals across those projects. We're made up of about 50 institutions and cover about 30 countries. We don't have a formal membership, so we don't exactly know who's involved in any particular time. And we have some infrastructure we developed. We rely quite a bit on our email lists from the beginning. So we have a big email list, and we have several ongoing threads and sublists to manage various projects. We do use Slack. I'll come back to that a little bit more in a bit. We use Google Drive for more flexible, fast-moving file management. And then when we have our projects where we want them to be, and we have the analyses done and the code done, and the data all formatted, then we use GitHub and OSF for keeping these things for posterity. Now to run through a little bit of the infrastructure. We have a website. You can have a look at it if you want, but it's simple. We try to keep it kind of simple and straightforward. We have various informative subpages, like one for each of our projects on the sidebar, and we generally try to keep this pretty simple, though. Again, we have been using Slack, but the ways in which we use Slack has changed quite a bit. When we started using it, it was free, and you could just use as many messages as you want. And as the subscription policies have changed, we no longer are able to do this. So we have a historic archive of all of our Slack data, but we don't use it for anything really like that we want to keep hold of, anything for posterity anymore, because we will lose the messages in 30 days. So that's required us to change our workflow a little bit. You can have a look at our GitHub. Again, you can see we have projects for all of the major published papers we have so far. Our pilot study, our first empirical study, and that review. The website is linked in here. And again, this is just freely available on GitHub. And here's a little snapshot of what we do on OSF. This is for Many Primates 3, which is the current empirical project we're running right now. This is another little bit more detailed page. Pretty straightforward, but you can see right now our pre-registration is already up and available in case you want to look at that to see how if you want to see the details of how we work on our pre-registrations. Now, I want to talk about the development process, because this is probably maybe the most important aspect of what we do and how is core to how we function. So from start to finish, in terms of our project development, firstly, we choose a topic, and we source our project topics from the membership. So at various points during Many Primates history, we have sourced projects. We basically ask our membership, hey, do you have ideas for projects, for topics, really, that you think would be of interest to the membership, and that we ought to learn something about by studying a big, wide cross-section of primates. So something that's really broadly applicable and important. And then people put together proposals in support of these topics. Those will get circulated to the membership. Membership will vote. And then that will give us whatever the most votes. We'll choose the topic. And then we'll request volunteers from the membership to staff our task force for that project. And we don't have any dedicated funding. We don't have any staff working on this. So everything that we do is done through volunteers. So these task force, they'll then explore the literature on the topic, first of all, and they will eventually develop a paradigm, an experimental paradigm, this is a challenge because the goal here is to create something that is simple, straightforward, doesn't require so many trials, doesn't require so much training, can be used with a wide variety of primates, and also builds upon the lessons that we may have learned in this area of the literature in the past. So it's a fairly high bar, but I think we've done pretty well so far. And then we get a lot of feedback, we have to go through a lot of rounds of revision. And when we're satisfied with the paradigm, then we pre-register the whole procedure, and we lists and other outlets letting people know they can collect data. And in order for the data collection to come off without a hitch, really, people will check out the protocol, see if they want to be involved, and then they will liaise with members of the task force. And so they will generally, at a various site, they will build a setup that meets the specifications that we laid out in the protocol, and then they will do a little bit of piloting, film it, and send it back to members of the task force, who will review the film footage and say, yeah, okay, this is what you're doing is exactly what we want you to see doing. This is fine. It fits within the degrees of freedom for our paradigm. And on the task force side, they will produce data sheets and various other resources that we try to make them as simple as possible so that we can just send the data sheets to these sites and they can fill them out. and send them back to us with the complete data. So once the data collection window closes after a specified period, then we will have all of our data. We'll carry out the analysis. Ideally, we will always pre-register before we carry out an analysis. And ideally, we will have written a code such that the data can just go straight into the code. We don't need to rewrite anything. And then data goes in, code already exists, data goes in, results come out. And with results out, we work on collaboratively writing the papers. And at this point, other individuals may get involved. Not everyone who's involved in many primates has access to primates, but we still like to, again, to maintain openness in the project so that anyone can be involved. And writing is a great way for people who don't have immediate access to primates to get involved. And so we write collaboratively with a bunch of people taking different sections of the paper. Okay. So what have we done through this? Well, so far, well, generally we conduct phylogenetic primate research. That's our main remit. We've done a few reviews like the one I showed you in chapters, and then we have some empirical projects. We have 3 that we've undertaken so far at various stages. And we're also working on some data linkage right now, including data for personality, demographics, and even welfare. But I want to talk to you a little bit more now about Many Primates 1, which is our short-term memory project, because it is a complete project. I can easily say the most about that. So in this first project, we wanted to look at primates' basic short-term memory ability. So we did it through a pretty simple paradigm, a cup paradigm, where the individuals are shown 3 cups, they're folded down, and they're shown a reward. They see that the reward goes underneath the cup, and then the reward is hidden under the cups. And there are 3 conditions, the 3 delay conditions. In the short condition, the board is just pushed forward and the individual can immediately choose the cup. In the medium delay condition, there's a 15-second delay. In the long delay condition, there's a delay. So this is just a basic test of if you make the individuals wait, how long can they remember where the reward is? So this is a few different sites in the picture, and I'm going to show you some video now from a couple of different sites. So here's the basic setup. The demonstrator shows this gibbon that there is this reward, shows it where it is, and then hides it. And then a timer starts. I think this is a long delay question. Yeah, this gibbon really gibbon knows where the reward is, and it really wants the reward, but it has to wait. Okay, time's up. Board goes forward. Gibbon can reach out and make its choice, and it chooses correctly. So while the gibbon eats the reward, I believe it's carrot, the demonstrator will get the board back into the neutral position and get set up and wait until the gibbon finishes eating so it can start the next trial. Okay, so it's finished. So the demonstrator shows new reward, places it, covers it up. Delay begins again, and the same thing happens. Okay, I'm gonna move ahead. Okay, so now we're gonna look at this basically exactly the same thing, just in a different setup. And so this is a good way of having a look at the kind of site variability that you get. Okay, so in this case, there's a lemur, but again, treat goes on the board, is occluded, delay now begins. And it's also interesting to watch how this lemur behaves differently from the gibbon. The gibbon was very focused and quite intent, and the lemur is a bit more spacey. But once the delay is up, though it takes a little time, looking around, this lemur does know where the reward is. So, okay. So I'm not going to talk too much about the results or analysis. I'm going to leave that for Bernhard. But the main upshot of our results was that the great apes have developed significantly better short-term memory abilities than the other clades of primates. You can read about this. The paper is published. Everything is out in Animal Behavior and Cognition if you want to learn all about it. I want to talk really quickly about some of the kind of lessons that we took away from MP1. Firstly, the start and end dating timeline. So when do we start data collection, when do we end data collection? Because there's quite a few factors that can influence this. Firstly, the development process of the experimental or data collection paradigms, whatever they are, that can take a while. The data collection can also take a while, but you don't want to box yourself into a particular timeline. It's good to be flexible about that because you may think you've done, as often is sort of the case with reviews. You think you've done a great job when you put together a paper and you've done an analysis and everything, but inevitably you get back reviews saying that you need to change some things. And it's exactly like that when we have these task forces that solicit feedback from our membership when we're developing these projects. We get more feedback than we think we're going to get, and so it means that it can take quite a long time to develop these paradigms. And so you don't exactly know when you're going to start, but when you do start data collection, there are all kinds of things that can happen. All three of the many primates projects thus far have been impacted by COVID in different ways. MP1 we didn't even have any data collection happening during that time, but still we ended up extending the data collection simply because we didn't know what kind of challenges people would run into or when people's schedules, when people would have the scheduling availability at their sites. So it's good to be flexible about these things if you can. Secondly, recruitment and participation. These are kind of two interrelated challenges. It is a challenge to get people involved in these projects, particularly as time goes on. There's always attrition issues to deal with. And as the projects age and the networks grow, people may leave the field, they may change fields, they may simply not have as much time to devote to the network as they used to, as their career roles changes. So it's important to think about the long-term health of these networks and not take them for granted. And data sharing, to say a little about this, which is that it's something worth thinking about and being careful about and making it clear the expectations that you have from the various people you work with at different sites. What data exactly do you absolutely need from them? And make those kinds of expectations clear in advance. So we had a few issues with this, small issues, but they were easy to resolve because and if you look at some of the MP1 data, you'll find that some of the individual primates, their names are there. They're fully identifiable, and that's fine for those sites. But some of the sites, there are anonymous codes so that they can't be identified, and that's fine because that kind of information isn't important. What is important is the various data from the trials that we collected, and we made it clear and managed the expectations that that's exactly the kind of stuff we needed in order to be able to do the analysis. Okay, so in summary, historic pressures in the field mean that there is much of the prior literature that contains bias, and it can be hard to know what to take away from older studies where design can be so variable and expectations very different depending on the field or the journal or what have you. And this is where replication and cross-site collaborations come in. There's no right or wrong way to do this. Every big collaboration must find its own feet, develop its own culture, and there's no way to please everyone either. I think a lot of us at Many Primates wanted and thought it should be obvious that every primatologist should get involved with it, but there are always going to be people on the outside. And that kind of thing, just like you can't ever make a project where everyone ends up being involved. And it's probably for best. It's good to have critical people on the outside as well. So for us, it's really important. I want to hammer this home again, that we maintain our quality and robustness of our work by following the principles of open science. That's replication, reproducibility, pre-registration, maintains our methodological analytic rigor, and also openness and transparency really maintain kind of the network and the trust within the network. Okay, that's about all I have to say for now, so I'm going to hand things over.
Liza, I cannot hear you. You're muted.
Sorry. Thank you. I just was thanking Drew for that talk and asking people to save their questions till the end. You can already start writing them in the chat if you have questions right now for Drew, and we're going to move on to Bernhard.
Okay. Start sharing my screen. So thanks, Liza. Thanks, Christian, for inviting me again to give a presentation. And I've been involved in multi-lab collaboration projects for the last couple of years, several of them. And to be quite open with you, it was often really difficult. It was a logistical and an administrative nightmare. So there were projects with starting from 2, but up to 6 or 7 collaborating laboratories. We are working in mouse research with mice. You need experimental licenses for this. And if there are 3 or 4 different countries involved and you have to get experimental license in all these different places for the same experiment, this becomes really challenging. So it is hard. And there are many aspects which makes it more difficult. You have to organize, you have to basically coordinate and talk all these other collaborators into really doing the study in the end, and they have to deal with all these administrative burdens. So these are all things which basically are rather scary, and therefore people are often very hesitant to think of multi-laboratory collaboration projects where data are collected at different sites. Another aspect which is something which is not very clear sometimes, what do I actually gain if I do a multi-lab study, and how can I analyze the data? What's the basically the additional benefit from having 2 labs involved? And one important aspect is definitely that by having several labs, you increase your external validity, because each, as we have seen in the previous presentation by Drew, each lab is doing things like slightly different. So there is variation between labs. Even if you're working with the same species, setups might be slightly different, and all this can lead to, might affect the outcomes. So having several labs involved is definitely a potentially very good tool to increase your external validity. So basically the chances that when your study will be eventually replicated somewhere else, the results still hold. This just as a start. Once you do
Bernhard, sorry to interrupt you. Could you just put it in presentation mode? Because now we see your outlines.
Yep. Okay.
And it's not new.
I'm giving away all the good stuff already. Now I can start in presentation mode. So, as I said, one more concern is how do I actually analyze the data? And there have been some interesting discussions. What do we do if I have 2 or 3 or 4 labs collaborating? Do we just throw all our data together? Is everybody analyzing the data separately? My idea for today is to give you an overview of the different ideas and also the discussion which is going on, with the motivation that if this becomes a little bit more clear, you see that actually there's not too much magic behind, and that there are several good options how to go on. And basically, by doing this, I hope I can convince you that it might be worth the effort. So this is what I actually already briefly hit on. So if you have 2 different centers, 2 different study centers or labs where you do an experiment, or even 3 or 4, you will get slightly different results in each lab, because labs differ in the ways how the animals are kept, or in the ways things are done. And from an ecological perspective, this is a concept well understood in terms of norm of reaction. So you have different environmental conditions, and these environmental conditions will lead to different phenotype values. You can have basically the special case that there is no big difference between phenotype values between environmental conditions. That's what you have in the on the left side. But you can also have a main effect of environment, so that between 2 different environments, you see differences, for example, in certain phenotypic values, both in a treatment and a control group. But the difference is the same, basically, at both between treatment and control is the same. That's the panel in the middle. Or you can also have an interaction between treatment and environmental condition. This is the third option on the right side, where depending on the lab, on the experimental center, treatment effect might be differently strong. And we must be prepared for all these three cases. And if we have just one lab where the study is done, we actually don't know about this. But if we replicate the study at a different site, or if we plan from the start to do it at different sites, we can actually make some statements about this norm of reaction. These are results from a study from Ivana Jaric, who worked with us here in Bern for a couple of years. This was a multi-center study with 5 different sites involved. So we ordered animals from one breeding facility, then we distributed them over 5 different labs just for rearing them. And then we made a battery of tests with these animals, but these tests were all done at our facility. So for testing, we had just one environment, which means standardized testing condition for all animals, but they had different rearing histories. And we found quite differences in, of course, the gut microbiome, but also development, then some epigenetic marks, differences in the behavioral and in stress responsiveness. So just keeping animals for a while at different laboratories or so can lastingly affect the phenotypes. That's the short message here. And this is a good reason why we want to do multi-laboratory studies. So how to analyze them? The data, if you basically make any experiments at 2 different sites. The easiest way is to say, okay, we have these 2 different sites, and each lab is analyzing we are analyzing the data from each lab separately. So we have a result for lab 1 and we have a result for lab 2. Now, this is possible. But it might not be the most efficient way. It has advantages. It's easy to do and to understand. So if we just have a control and treatment group, means that each lab manager has basically just to do a simple t-test. The t-test is very robust. It's very well understood and easy to perform. So you get rather straightforward result. The problem is that if you do this at 2 or 3 labs, you test multiple hypotheses, so you have to correct for multiple testing. And if you analyze, basically make a test at each center, this also means that you have to power the study at each center in a way that you can actually run the test at each center, which means in the end, you're ending up with a much larger sample size. Also, there might be difficulties in the interpretation. If you have differences between centers, you can visually see them, but because you analyze the data separately and made no joint analysis, you cannot really make any statistical inference about differences between centers or main effects or interactions. So for this while I'm already talking about correcting for multiple testing, now there is one common misconception that multiple testing correction is only needed when you do a lot of tests. And just to mention, this is not the case. So even with just 2 or 3 labs involved, correction for multiple testing is absolutely essential. In fact, the largest difference in your threshold value for or in the correction factor for your threshold value is actually the largest step is when you go from 1 lab to 2 labs. Then there is 1 from 2 to 3. Whether you make 6 or 7 tests or so in statistical terms for the correction method for multiple testing, that doesn't make a big difference anymore. So it's really, as soon as you do 2 or 3 independent tests, then you have to correct for multiple testing. This is definitely a disadvantage of making separate analysis. The way forward that was suggested already by Ronald Fisher in the 1930s is the so-called 2-way ANOVA design, where you include center then as a factor in your analysis. So you have treatment and you have center, and you can also include the center by treatment interaction. So the question whether treatment effect differs between centers. Yeah, this is a very classical approach and works very good. It works best if you have a clear orthogonal design, which means you have a cross design, so you have control and treatment groups in both labs, and you have a balanced design, which means you have the same number of animals in both labs and for both treatment and control group. If you have this, you have a very powerful test to detect both main effects of the treatment, but also you can look at differences between labs and interactions between the environment and your treatment variable. So the big advantages of the two-way ANOVA design, it's still very efficient. So if you have two labs, that does not mean you need twice the sample size than if you have just one lab, but only a very modest increase in the sample size will give you a very powerful study. And the advantages, you can have, of course, you see the main effect of treatment, but you can also see the main effect of the environment or the interaction between treatment and environment, which makes your results more generalizable. The disadvantage of the ANOVA approach is that you have, if you do a classical ANOVA, you have certain distributional assumptions. And if you have more than two labs, you might need post-hoc tests to then later see where the differences between the labs are. And because you also have these interactions, the interpretation is a slightly more complicated than if you have just a single t-test. Now, another option is a pooled analysis of all the centers you have. So in this example, just two labs. So what you do is you basically pretend that you do not know that the data points are coming from two different labs, and you just throw them together and make a single t-test again. This might sound quite strange. This is actually the approach which was rather dominant for a long time. Until the 80s or so, you will find it in many research papers in medical science that if they have many centers, they just pulled the data from those sites. It has disadvantages. One of the disadvantages is that it increases your estimate for the variance. So imagine you have two labs with and in fact, the two slightly different underlying distributions. So an effect size of 0.5 in one case and a standard deviation of 0.6. And in the other case, an effect size of 1.7 and a similar standard deviation of 0.5. Now, if you pool them, you get a distribution like this, and you say, okay, this looks still like about a normal distribution for me. So I just pretend it's normal, and I'll just throw this into a simple t-test and get my results. You can do this, but the result is that because of these two slightly different underlying distributions, which have rather narrow standard deviations, you arrive at joint distribution, which has a rather large standard deviation. So this is one disadvantage. So inflation of the variance, if you do not account for the underlying structure in terms of two labs in this case. In terms of statistical power, this means with this case where you do include the information about these two underlying labs, you would, in this specific example, arrive at the power of 0.96. But if you just pool them and ignore the structure, then your power goes down a little bit with the same overall sample size. Okay, this is a small inconvenience. However, people sometimes argue that it's not that big a problem because in laboratory settings, very often we underestimate the true variance anyway. So having a slightly inflated variance here is just basically countering this effect. The bigger problem is if you pool your data, you're not able to detect interactions between your treatment and the specific environment. So this example here, where you basically have a statistical interaction, an effect in one direction in one lab and the other direction in the other lab. If you just pool the data and ignore lab membership, you do not see this effect anymore. If this happens, then and you interpret this just as the absence of any effect, then you're really missing something. And the third problem that can happen is confounding, specifically if you have unequal sample sizes, like in this example, where you have, of course, a main effect of center of the environment, and you have unequal sample sizes. This could lead to an effect where then, if you pool the data, you find actually a controlled treatment effect that doesn't exist, but it's just a confounding effect due to lab differences. Now, for these reasons, pooled analysis have been criticized heavily for a long time. Still, they are easy to do. That's why they have been done in the past. You can do them with very small samples. And if you can be sure that you do not have interactions, and if you have an orthogonal balanced design where you have equal sample sizes in both groups, it might still work. So and this is why even today you sometimes see people doing this. And yeah, we have in a simulation study, we have tried out what actually happens if we take real world data and basically try out either pool the data from different studies or just analyze data from studies separately. And so these are these two scenarios. In this case, we say, okay, we take real world data from different studies with mice. We always take data from 3 different labs. We pool them. And then we just ignore this lab membership and make a simple t-test and calculate our statistics. What do we get out? And the interesting thing is, so that it depends on the sample size, of course, and on the numbers of lab participating. So we simulated it for 1, 2, 3, or 4 labs. Now, I just want to draw your attention to, do I have a laser pointer here or something? Yeah, laser pointer. To this graph here. So what you basically see is that if you pool data from, and we just focus on the blue data points, which is an overall sample size of 12. And if you pool data from different labs, what we see is an increase in the width of the estimated confidence interval. This is the effect that I mentioned before. But because of the wider confidence interval, it's getting actually more likely that the study that the outcome of the study replicates if you compare it with new data, because broader confidence interval means also it's likely more likely to be hit by another study. And when it comes to false negative rate, you see different effects depending on the sample size. So everything else being the same. So for very small samples, pooling data actually increases the false negative rate. But if the sample size is large enough, then pooling data can actually lead to a decrease in the false negative rate. So this is just to mention that just plain pooling of data is not necessarily detrimental, but can have actually advantageous effects as well. The final analysis tool that is becoming more and more popular is the generalized linear mixed effect model, where you're adding the center as a random effect. This is in principle a very good approach. So you say, okay, these 3 centers that participated in my study are a random sample of many, many centers who could have participated in this study. So I treat center as a random effect variable. Now, in principle, this works. Practically, if you do this, you will very often end up with warning messages like this. If you're working with R, you might have seen them. The problem is that generalized linear mixed effect models were originally developed for large data sets, and large data sets is something with 600 entries or more. So if you have just 3 participating labs and 20 or 12 mice per lab or 20 mice per lab, you have a very small sample, small for a generalized linear mixed effect model. And the result is that very often the algorithms behind these models might find it difficult to actually estimate the parameters, and then you get these warning messages. And then it's actually uncertain whether the model estimate is in any way good or not. So that's the main problem with the generalized linear mixed effect model. And another thing is once you get these warning messages, you have different ways how to deal with them. So you can look for different fitting algorithms. You can play around with basically the quality thresholds. You can play around with quite a lot of things, or you can include more variables or exclude variables, which in terms, well, if you actually want to pre-register your study, makes it more difficult. And specifically in biomedical research, where you should really have a straightforward way to analyze your data without any researcher's degrees of freedom, how you can fiddle around with the analysis until you find your preferred result. So the linear mixed effect models are a bit tricky in this respect because it's a different approach to do statistics. You first make the model, then you check whether basically also with diagnostics, you check whether the assumptions held. If not, you revise your model and you revise it over and over again. So that's a bit of a disadvantage of this approach. So in general, it's a very powerful tool. It's the Swiss Army knife. You can include everything, but they are more complicated. You need larger sample size, and you very often need diagnostics and adjustments to get to the final result. So to summarize these different approaches, to over and over, if you have a simple design, just control and treatment group, and you have a few centers, then that's definitely the most preferred way to analyze your data. You can go for more complex mixed effect models, but be aware they are tricky and they were originally made for large samples. And yeah, you might run into troubles there. Pooled analysis have been popular in the past, have been seen critically, but still exist. And there are also some, not too bad reasons why under certain circumstances they can still be useful. And separate analysis is, I put it on position 4, is maybe the least effective way to analyze your data, but maybe there's one or the other case where it still makes sense. So none of these methods is perfect. All have their plus and their minus, the drawbacks, the advantages, which means, it's really depending on the case to case. Specifics, which of these approaches might be the most appropriate, but they are all under certain circumstances feasible. That's basically what I wanted to say, and I hope this made some sense, and I would be really pleased if there are critical questions.
Wonderful. Thank you so much, Bernhard. Thanks to both of the speakers. And you guys, thank you for being perfectly on time as well. So we have at least 10 minutes for questions. And you can, I don't know if I can see everyone, but let's actually stop sharing screen maybe. We already did that. So I'm gonna just try to see you. If you raise your hand, hopefully I will see you, and otherwise you can write in the chat if I'm not seeing your hand. And I'm gonna start with some questions in the chat already. First two for Drew. And I'm just gonna summarize them a little bit. So yeah, an interesting question about, I think, based on the methods in the video you were showing us, I think it's a really good kind of framework to pick a simple design that's easy to implement across these labs, and we saw that there. But the Berta is asking, so how do we actually know that the primates are using the same kind of method to solve it across the different labs? So specifically, she's saying that it looks like in the lemur example, there's maybe olfaction being used to determine where the reward is. So how do you know that the subjects are actually reacting consistently in the same way, or just using the same kind of cues?
Yeah, it's a good question. And like the bottom line is kind of no way to do that perfectly with just one, particularly with just one experiment in this kind of situation. In this case, this is kind of a good example because these videos came from the piloting phase. That's where they come from. So when we observed this happening, I don't remember exactly since this is from like 8 years ago or something like that. But when we looked at this video, we observed this. And so I can't remember exactly what we did what we recommended in this case. If it was like use a very non-pungent treat, see if that makes a difference, or even use a kind of interfering odor on all of the cups and the location so that odor couldn't be used as an extra cue. But this is all part of the process. This is what kind of like I like it as an example because they give us these the sites will give us these videos and then we have the chance to give them a little more information about what we noticed to at least one round of feedback to try and make it all a little bit better.
Great. Thanks for that. And one more question for Drew. And this is, I think, somebody who came in later. So you'd mentioned that Many Primates isn't directly funded, but this person is asking, Laura's asking, so are the participants, are they all involved specifically through universities or institutions, or is there some kind of process for how people can get involved in the research, or really can anyone volunteer and become part of one of these projects?
Yeah, it's a good question. I mean, basically it's just about anyone who can get involved. We don't, again, we really prize our openness, so we don't set any particular barriers in place to try and stop people from getting involved. That said, it involves working with primates. So it's not like you can get that many citizen scientists, like if you were working with dogs and people wanted to submit data from their dogs. If you have access to primates, you know someone, you have some kind of pathway to get access to primates. So there's a bit of vetting there already. But the people who tend to volunteer for this, like to collecting data, there are people who do undergraduate master's projects for their thesis. They'll collect data, they'll do that. People will do it as part of their PhD. Postdocs, when they have some extra time, they will often work on this. But the funding, if anyone has funding, it's never directly for these kinds of projects. It's everyone taking a little bit of time out of their schedule that they would otherwise have to work on these kinds of projects, whether it's collecting data or whether it's writing or paradigm development or any aspect of it.
Great, thanks. I don't see any other questions in the group, so I'm going to jump in and ask Bernhard a question. Oh, there's going to be a question, but I really want to ask Bernhard, so I'm going to get this out before, and then we'll see the next question. So comparing because I just did an analysis, kind of trying to deal with a lot of the things you brought up. And you said in the ANOVA, you would have the institute or center, the different lab as a fixed effect predictor. But if you move to a GLMM, interestingly, it would become a random effect. And first of all, I'm questioning that logic. Why wouldn't it still be a fixed effect in your GLMM? Or when is it safe to put the institute as a random effect? And then either way, whether it's a fixed effect or a random effect, what I came across and that I think a lot of us will deal with is you don't just have one factor there. You have the different location, and then you have these different, what do you call it, environmental norms, or the norms of reaction that could have to do also if there's breed differences across the different locations, or differences in how the personnel are handling there's so many different factors that go into this institute predictor. So it almost felt at the end very unsatisfying because we could see there's institute differences, but we didn't actually know, are they breed differences? Are they management differences, or are they really, something else? And how do you deal with that in these studies?
Okay, that were a lot of questions. The
Sorry.
first question, why in the mixed effect model the lab is a random effect and not a fixed effect? If it's a fixed effect, then it's just a 2V ANOVA. Then you don't have a random effect, unless you have other random effects. But in the simple case where you have just treatment and lab, treatment is for sure a fixed effect. If lab is also a fixed effect, it's called a 2V ANOVA or linear model. If lab is a random effect, then it's the mixed effect model. As to the question when to treat lab as a fixed or random effect, in principle, depends on the number on several decisions. So there's this one idea that if you say lab is just a nuisance variable that I included in the model because I want to be sure that I acknowledge that there is that there are different labs. Then it's just a random effect, but I'm not really interested in the size of the random effect. I just want to be sure it's in the model. If I really want to estimate the parameters between and behind this factor, then I need a lot of levels, which means a lot of different labs. If I have just 2 labs, I can of course calculate the variation that I have between the labs, but with standard deviation estimate based on 2 is not very precise or basically meaningless. So there are different opinions. Some say least number of levels for a random effect should be 6. Others say 12. Others say 20. Others say 42. But it's basically just some numbers people made up. So this is if you have just 2 or 3 labs, you might be on the safer side to treat it as a fixed effect. Actually, well, it's also not wrong to do a mixed effect models with just 2 factors. It just basically means that the mixed effect model boils down to basically the solution you would get with the fixed effect model anyway. That was the first part. The next part was you have so many factors. Yeah, that's the good part, and this is why it might actually make sense to model lab as a random effect, because this reminds me a bit of Fisher's infinitesimal theorem, where he says, okay, why are so many biological traits normally distributed? Because you have thousands of different genes, each gene making a tiny contribution to the final phenotype, and this is why you get this wonderful bell-shaped curve. With a lab, you have the same thing. You have 1,000 different differences between labs, as you say, the mood of the caretaker, the feed, the light conditions, the heat, and all these things can have a small contribution to the overall phenotype. And because you have so many things, it actually makes it more likely that labs can really thought of being a random, normal random variable.
That's a really good perspective. Thank you. That helps a lot.
And basically, okay. Lab is basically subsuming all the difference that you have between the labs, from the caretaker to the feed and everything.
Great. Okay, we have a lot more questions, so I will stop asking questions now and go back to the feed. For both people, how do you keep inertia high? I mean, I think he means low. With all the other academic and non-academic things trying to get our attention. So basically, the argument is that these multi-site studies, it's kind of this democratic principles of open science and sharing, but it's not really rewarded so much in the typical academic settings. So how do you guys? Do you have problems with finding the time and the resources to do things like this? What motivates you to keep working on these kinds of projects? Or do you find support within the more typical institutional framework as well for this? And anyone can, whoever wants to answer that.
I'll chip in first just for a little bit. Yeah, it is difficult. The incentives don't align very well with big collaborative projects. Because of the way funding structures are, and that they're still very focused on single PI-led grants. And they take a long time, which can also be an issue. They're not fast. They're not quick and rewarding. So it is a challenge, and it's changing. People are starting to see that that bigger cross-site collaborations are a better way to do research. So that is helping, and the incentives are gradually changing. They're kind of working their way up through the institutions. But it's also something worth thinking about, because everything changes, and with this many people, when you have the more people you have involved, the more degrees of freedom there are that way as well. So it's worth thinking about how you are going to maintain a network over the long term, and thinking about it quite broadly and quite deeply as well. Yeah, that's about it for now.
Bernard, did you want to add anything to that?
I mean, for one, there are now different funding schemes which are dedicated to support, multiplayer projects. So there's definitely motivation there to, I mean, if there is money, you will find researchers who want to have this money. So you team up in order to get to one of these pots. That's definitely, something that works. And I mentioned at the start that it's actually rather tedious to administrate these projects, but on the other side, it can be also socially rewarding, specifically if you work together with people you like and you get along well with, and it's good. It's a good way to make good friends. So yeah, should also mention that aspect.
Nice. Yeah. And I think that overlaps with the goals for the LIFT kind of framework and getting involved in something like these cost actions. So Bernard had mentioned some challenges analyzing data across sites. And this person is interested in knowing, could you both share some other challenges that you encountered managing cross-institute collaborations?
I mean, really aligning the protocols is sometimes really difficult. You have certain things you cannot align between centers if they're in a different country. Sometimes they have slightly different reagents, and it then turns out that they have to be different because there's different legal regulations. Also with animal husbandry and animal procedures, there are sometimes differences between countries, or even in Switzerland, between different regions of Switzerland, where you basically have to follow your local regulations. So, and then there are also always these unforeseen difficulties where you didn't think that at the other lab they make it differently, and then just turns out they do it this way and we did it this way, and then you have heterogeneity. But I mean, the good thing is then you start thinking about it, whether this is really relevant, and you start discussing this, and then you're talking biology, because then you really get to the point to be more specific. Okay, what's really needed? What are the essentials? And what is just irrelevant anyway.
Thanks. Drew, did you want to add anything?
About the challenges across sites.
Yep.
Yeah, kind of hard to how to narrow it down into one or two, particularly with the data analysis. The compartmentalization of the process certainly helps. So like our analysts are just looking at the data and they don't know any better if there's like issues in the data when they get it. They're just writing the code based on what they expect. And that can help. But for instance, like the delay conditions were added later on. There was only a short and a long delay in the original version of this, and we added the third delay condition in the middle later on, and which then you have to update pre-registration and so on and so forth. And so that's something I mentioned before. I think what really helps is if you can be flexible. And there are various aspects of open science which like it doesn't seem like they support flexibility, like registering every advance. Like you want to reduce your researcher degrees of freedom through mechanisms like this, but you can kind of build in the opportunity to be flexible if you plan and you think about this in advance. And if you just document the ways you change things, there are even guides on when you deviate from a pre-registration or when you deviate from a registered report, how do you do it? Because sometimes you just have to do that, and you have to accept that the perfect should not be the enemy of the good. And that can happen. I mean, that happens with data analysis and basically every project I've ever been involved with, I think.
Great. So no more written in questions at this point, and we're actually a few minutes over. I think if there's other burning questions, I'm sure the speakers would be happy to answer them. And otherwise, I just want to say I think there's really a lot that we can take away from in thinking about how to try to expand our perspectives, and also already within the cost action, there are lots of possibilities to do more of these cross-institute, cross-lab collaborations. So I hope we're already trying to take advantage of that more. And we have, both of you cited some really nice examples from your work that we can refer to also for more detail on how this is done. So that's also really helpful, the resources that you brought up. And yeah, just want to thank you for your contributions and for inspiring us with these kinds of topics. So thanks a lot, both of you. And yeah, we'll see you guys at the next seminar.
Thanks so much for the invitation.
Okay.
Thanks for having us.
Thank you very much.